Hi, my name is Tao Wan and I work in the STOS group as a research assistant following my PhD study at the School of Chemistry at Southampton University. So my current project involves testing chemicals to see if they can penetrate the skin cells membrane and get taken up by live skin cells. So the aim of our research is to try to improve the current treatment of skin cancer by creating compounds that can be taken up by cancer cells quickly, effectively, and also can kill them selectively. So here are some samples of porphyrins that we make in this chemistry lab. So let's go and test them out in the School of Medicine. So to have the skin cells hackets ready for the experiments, I had to grow them in the incubator chamber for a few days, a process which is also known as cells culture. And now let's check how well the cells will grow under the microscope. So here you can see the image of hackets. Hacket cells are very sociable cells. They like to be together and next to their neighbor and form layers of hacket colonies. That's why you can see a really nice confluence of cells under the microscope. So the next step is to harvest the cells, and in order to do that, I need to use an enzyme called trypsin. So this will help to break the protein bonds between the cells and the bottom of the flask. And then I will need to leave it in the incubator to work for 5 minutes. Here you can see upon the addition of trypsin, the skin cells start to come off the flask and separate it from their neighbors, as these individual cells seem to enjoy having a swim in the solution. Then the cells can be suspended in solution and ready to be transferred into petri dishes and left in the incubator overnight for the cells to grow. I'm going to pipette the porphyrins into my cell samples and wait for a few hours to see if the porphyrin can penetrate through the cell's membrane. Because porphyrins are fluorescent, which means they emit light in the visible region. So when they get into the cells, they will make the cells glow, and the image captured under this fluorescent microscope can provide evidence for a successful cells transfection. So in this photo, you can see the porphyrin has successfully penetrated into different individual skin cells at different rates. Some absorb more porphyrins and hence glow more intense than others. It seems that the porphyrin has been taken up by the cell's cytoplasm. For cells that fluoresce with porphyrin, I'm going to shine LED light on them to check whether the porphyrin actually induces killing of these modeling skin cancer cells upon being excited by the light. So the results show that compared to the control sample of cells without porphyrin, the cells with porphyrin show a dramatic decrease in the number of cells. This is indicative that the porphyrin has induced cells death upon exciting the molecules with light. So eventually we would like to apply our research from the cells model onto the skin's tumor to help fight skin cancer. This project in particular helps to reinforce that chemistry as a subject is very interdisciplinary and can have many real-life applications, such as in healthcare.